Hi everyone, I'm Aldric. I'm a physics PhD student at Brandeis, and I'm here today to talk to you guys about who I am, where I'm from, and how I got to do the research that I'm doing today. I'm from India, and the most famous monument in India is the Taj Mahal, and that's what you see on the top left. And the bottom two pictures are here to celebrate what a secular and diverse country India is, with numerous religions and a huge number of languages spoken by its people. The other pictures are pictures that uh, relate to my research, and I'm going to be talking to you guys about that in the next few slides. I'm from the state of Tamil Nadu, which is the southernmost state in India, and my city is right in the center, and it is Trichy. And like all cities in India, it's famous for a lot of temples. The most important one being Srirangam, pictured here, right by the river of Kaveri, which runs through my city. We also have this fort called Rock Fort, from which the nickname of our city comes, which is Rock City. And some of the buildings there were built in the first century. On the right are some of the food that I really enjoy from my place. First is filter coffee, which is essentially coffee with milk and sugar, and it tastes amazing. And on the bottom is a traditional banana leaf lunch or dinner, where they give you rice, a bunch of different curries, snacks, and vegetables, and you eat it with your hands. And I did my high school in Trichy, and my school was Montfort School. And while I was in school, I had to do a bunch of different subjects as part of my curriculum. But all this time, I had to also think about what I'm going to do in my higher studies when I was going for my undergraduate. And I was going to do that for the next 5 years of my life. So I had to choose what I really loved and do that. And when I looked at Tamil and English languages, languages were never one of my strong points. So I was never going to do a degree in language. Chemistry, well, my parents were both chemistry graduates, and they talked about chemistry a lot at home, and I was really bored with it, and so I'm not going to do chemistry. Biology, well, a very humane reason for me not doing biology was my teacher. I absolutely did not like her. I just did not, I couldn't make myself study biology in school. And so, not for my undergrad. History and geography was a lot of reading, writing, and remembering factual information. And that was never my strong point. So I decided not to do that. Coming to some of my favorite subjects, and first and foremost amongst them, physics. I really loved the subject because I could see what I was studying about. One thing that really fascinated me were pendulums, and the fact that you can write equations to describe their motion um, was something that blew my mind when I first saw it. And that's how I got interested in maths from physics, and geometry was one of my favorite subjects in mathematics. I also loved computer science. I loved coding, uh, and school gave me a sneak peek into how coding would look like, how writing a code in C would look like. Last one, soccer. Well, that's a different story. I really love soccer. I absolutely love soccer, but for the life of me, I can't play it. So I was never going to do it professionally. So it was decided that I was going to do physics because physics also has maths and computer science built into it. So to do my undergraduate, I traveled to the biggest city in my state, which is Chennai. And just like any big city in India, it's extremely crowded, and there's a lot of people, there's a lot of vehicles. But the good thing about the city is it's right by the seaside. It has a lot of beaches, and the fast food in the city is just amazing. And the city never sleeps. so you can get a snack at 3 a.m. if you want. And right in the middle of such a busy and dense city was my school. And the absolutely best thing about my school was its wildlife. It was carved out of a forest, and where I used to stay, there used to be black bucks and spotted deer just roaming around. School can sometimes be very stressful, and being in an environment which you like and love is very important for your mental health. And that's exactly what my undergraduate school did for me. While I was enjoying my campus, I also had to figure out what specialization I want to do, what uh, specific thing in physics that I wanted to do. So first, the hype was quantum computers. When I heard quantum computers, I was just fascinated by them. Just the promise and prospect of what they could bring to our like computers was just mind-boggling. So I joined forces with one of my favorite teachers to read a few chapters from this very basic introductory book on quantum computation. But soon I realized that this subject was not for me because I couldn't relate with the abstract math that was involved in the subject. So I went ahead with something that was completely different. This was a statistical physics problem called percolation theory. This whole theory works on how a two-dimensional or a one-dimensional space can be filled with dots or sticks. And while I was doing this, I taught myself how to code. I also taught myself how to do plotting, and I absolutely enjoyed that. But at the same time, I also came across this particular video of a white blood cell chasing and engulfing a bacteria. White blood cells are a type of blood cells that protect you from infections. And just the fact that they can just chase around a bacteria and do that really fascinated me. One other video I saw was the one below, where uh, the experimentalist is introducing food or chemoattractants, as they call it, into a dish with a bunch of these cells, and these cells immediately flock towards the food. Somehow the cells all over the place know that there is food there, and they start moving towards that. This really fascinated me, and I wanted to work in this topic. So I used my experience uh, that I've gained in the last few years to mathematically model such movement by the cell and also simulate them. While I was doing this, it was pretty clear to me that I wanted to do a PhD in physics. Very specifically, I wanted to do research in biophysics. That's when I came to Brandeis and I met my two mentors, Yane and Bruce. And these guys are really good friends and they do science together. Yane is a theoretical physicist, while Bruce is a cell biologist. And just one conversation with them, and then I decided, this was the problem that I'm going to work on. Well, what is that problem? Well, in the next few slides, I'll be telling you guys more about that problem. One fact that really amazes me every time I look at is the number of cells in your body. Your body has about 15 trillion cells, which is about 1,000 times more than the number of people in the world. And if you look at a textbook picture of a cell, you would see something like this, where you have a cell with a lot of organelles. But the one I am specifically interested is called the cytoskeleton. Well, what is a cytoskeleton? What is it made up of? Cytoskeleton is made up of actin filaments and microtubules. In this image, you can see actin filaments in red and microtubules in green. As you can see from the picture, the cytoskeleton components are present all over the cell. These guys give the cell its shape and structure. Not just that, actin filaments also help cells to move from one place to another. So cytoskeleton is not just important for giving cells their shape. They also do a bunch of other things. Well, on the left is a single-celled organism called Clamidomonas, and these guys use flagella to swim and go from one place to another. And these flagella are made up of microtubules, which I mentioned in my last slide was one of the cytoskeleton elements. And if you look closely, these two flagella are approximately of the same length. And this length being same is pretty important for them to move from one place to another. On the right is a structure called Stereocilia, which is present in your inner ear. These are actin-based structures which have this specific staircase-like structure where the ones in the front row are shorter, and as you increase the row, their length also increases. This staircase-like structure is very important for their function, and their function in your inner ear is to convert sound waves into electrical signals that your brain can comprehend. So these cells know how long these structures have to be and build these structures accordingly. So how do they do that? That's where my model organism, which is budding yeast, comes into the picture. It's a single-celled organism, and it's very easy to manipulate genetically, and that's exactly why I work with these cells. These are called budding yeast, just like the yeast that's being used to make bread or beer. And they reproduce by a process called budding, in which a mother cell, which is the bigger one you see here, puts out a bud or a daughter, which you see here. And to make this bud, the mother has to transport nutrients and resources from its compartment to the bud. For this, it makes these actin cables, which start from around where the neck, from around where the bud is, to the rear of the cell. And again, even here, the cell knows that it has to make them of a specific size to have efficient transport of resources from the mother to the daughter. What is more interesting is that these cytoskeleton elements are not just made and left. They are constantly being made and remodeled. By remodeled, I mean they literally get destructed, like the cell removes them and then makes them all over again. So these guys are always under construction. They also have these motor proteins which walk along them, which I won't be going into in this talk. Another thing that is very interesting about these structures is that they are made up of small subunits which are nanometer in length, just like your Lego building blocks. And the cell puts all of these guys together to make a beautiful structure, which is in the micron scale. And my research aims to find out how cells do this. For this, I use cell biology techniques to fluorescently label or genetically manipulate my cells, and I image them using fancy microscopes, which cost like $1 million and which enhance you to take pictures that are so cool. In addition to this, I also write down equations for my theory side. I test these equations and models in my computer simulations. So the major takeaways from my talk are 3 things. My biology teacher won at the end because I'm doing biology now. 2, you have 15 trillion cells in your body. That's a huge amount of cells, and all of them have something called cytoskeleton in them, which is made up of microtubules and actin. And the various examples I showed you before, cells know how to make these structures of a specific size. And my research aims at finding, how they do that. Thank you for listening to my talk. I hope you all enjoy science as much as I do. Thank you.